There are several different types of transport mechanisms available for transporting molecules across cell membranes. These are all summarized here on the slide. I will briefly review each of the different types of transport, and while doing so, I will point out whether the processes are passive. This just means that the molecule is being transported down a concentration or electrochemical gradient. So in this situation, you would not need any additional energy source because you're going with the gradient. The second type of process would be active processes, where the molecules are being transported against an electrochemical gradient. In this case, because we're going against that electrochemical gradient, the process will require some type of energy source, and we'll go through what each of those are. So we'll start off by reviewing the simplest case, which is our simple diffusion in the upper left-hand quarter. In this case, the molecule that's being transported is a nonpolar compound, so it's able to readily traverse through the cell membrane without any assistance, and this process simply occurs going in whichever direction is favored by the concentration gradient. So obviously this is a passive process. Next up is facilitated diffusion. Again, we are going to be transporting the molecule down an electrochemical gradient. In this situation, the molecule is not nonpolar enough to simply diffuse through the cell membrane, so it's just going to use the protein as a way to just guide it through that hydrophobic core of the cell membrane. So this is also a passive process that does not require any additional energy source. The other types of passive processes that there are would be our ion channels located down at the bottom of the slide. These channels transport the ions down the electrochemical gradient. Therefore, they do not require an energy source. In ionophore mediated transport, ionophores are, you can kind of think of them as little portable channels. So they are, again, going to kind of bind the ion and carry it across the membrane in whichever direction is preferred by the concentration gradient. So those four types of processes are passive, meaning we don't need any additional energy source. In contrast, there are two types of active transport. So in this case, both of these cases, we're going against the concentration gradients. So our two types of active processes will be the primary active transport. In this case, we are going against the electrochemical gradient, and this energy is going to be provided by ATP. And I want to point out, in this case, when we're using ATP as an energy source, we are hydrolyzing the ATP to form ADP in an inorganic phosphate molecule. This is very different than, say, an ATP-gated ion channel. So if you have an ATP-gated ion channel, that is not an active process because you're not actually consuming the ATP. It's just the presence of the ATP can trigger the opening or closing of the gate. So in the active primary transport, the ATP is hydrolyzed or consumed, and therefore that is an active process. The second type of active transport is secondary active transport. So in this case, we are going to use some secondary ion gradient, usually, to provide the energy needed to transport our molecule across the membrane. And we'll take a look at some of these specific examples of these processes in a moment. The other thing I wanted to point out was saturation kinetics. So if you are looking at something that is a simple diffusion process, such as indole crossing the cell membrane, again through simple diffusion, you see we have this linear kinetics. So as long as we increase our concentration of the indole that's available, we will increase the rate of transport. In contrast, if you look at the curve here for glucose, this is exhibiting what's called saturation kinetics. And that is initially while we increase our concentration of glucose, we get an increase in the rate of transport. That is going to saturate. So you see this plateauing effect because you do reach a point where the enzyme can no longer, or the transport protein cannot move any faster regardless of how much substrate is present. So looking at our facilitated diffusion process, this is going to exhibit saturation kinetics. This is really just a quick refresher of some of the terminology that we'll look at. So uniport here on the far left means we're simply transporting one solute across the membrane. Doesn't matter the direction, so just a single solute is being transported. We can also then have what's called co-transport. With co-transport, we're transporting two different solutes through a transporter. And those two solutes can either be transported in the same direction, such as our example in the center here, and that process is known as symport, or we can transport those solutes in opposite directions, and that is termed antiport. So this just tells you about the number of molecules being transported as well as the relative directionality. What you should be able to do is look at a transporter and be able to describe the process that is occurring. So in this model, we're looking at glucose transport through one of the GLUT transporters. So this is an example of facilitated diffusion. It is a passive transport process. We do not require any outside energy source. The concentration gradient of the glucose provides sufficient energy for that process to occur. This is also an example of uniport, since we are just transporting one solute molecule across the membrane. And this process will exhibit saturation kinetics, as can be observed in the graph here where you're looking at glucose. The other type of transport that can occur is active transport. So with active transport, we are moving a solute molecule against its concentration gradient. The first type of active transport is known as primary active transport, and this is shown on the left-hand side. With primary active transport, we are going to be deriving our energy to move solute 1 across its concentration gradient by the hydrolysis of ATP. So ATP is our energy source in this example. The second type of active transport is secondary active transport. In this example, we're going to be using the ion gradient of S1 in this example as the energy source in order to move solute 2, shown there in green, across the membrane against its concentration gradient. So you notice in this example, S1 is going with its concentration gradient. So that will be providing the energy that we need in order to move S2 across the membrane against the concentration gradient. And so we can look at some specific examples here of transporter processes. On the left-hand side, we have the sodium-potassium ATPase. So with the sodium-potassium ATPase, this is going to be an example of a primary active transport. What you can see is that we're moving two ions. We're moving both 3 sodium ions and 2 potassium ions, and those ions are being moved against their concentration gradients. And therefore we do need an energy source. And that energy source is going to come in the form of ATP. Again, it's the hydrolysis of ATP that provides the energy. What you should also notice about this primary active transporter before we move on is that you'll notice the sodium and potassium ions are moving in opposite directions. So this is an example of an antiporter. In our example on the right-hand side, we have the sodium glucose symporter. What we're doing now is we're using that sodium ion gradient provided by the sodium-potassium ATPase. So we have a very high concentration of sodium outside the cell. We're going to use this sodium ion as the energy source to move glucose into the cell. So sodium moves across the cell membrane down the electrochemical gradient, and it's going to bring along with it glucose, which is moving against its electrochemical or concentration gradient. So this is an example of secondary active transport. And you'll notice that in this example, the sodium and the glucose are moving in the same directions. So this is also known as symport. Now with these secondary active transporters, we'll take a look at what happens if you affect your energy source. So here we're looking at another example of a secondary active transport process. This is the lactose transporter. With the lactose transporter, it is going to use the proton gradient in order to, that'll provide the energy to transport lactose inside the cell. And that's very similar to the sodium glucose symport we were just looking at. Well, the proton pump is responsible for providing the proton gradient that's used as the energy source. So if we in any way inhibit the proton pump, if you look at the graph here in the bottom right-hand corner, the red shows just our active transport process. Notice you do see the saturation kinetics occurring in this process there. If we either add a small molecule inhibitor of the pump, such as cyanide, or if we mutate the pump to something to a basically mutant that has lower activity, what you can see is that our lactose transport is going to be decreased. So that's what you're looking at in both of those examples. So if you do anything to alter production of this proton gradient, you will inhibit your lactose transporter. So no gradient leads to no transport through the secondary transporter. The last type of transport that we'll look at is transport through ion channels. So transport through ion channels is much faster than transport through some of the facilitated transporters we were looking at, like the GLUT transporters or the symporters, antiporters that we were looking at on the previous slides. So with the ion channels, what we're going to need to do is we start off and we have a hydrated potassium ion in this case. The water molecules are going to have to be removed from that potassium ion, stripping it down to just the naked ion that combined in the channel, and we have these alternating ion binding sites. What happens is there's going to be charge repulsion between the neighboring potassium ions that's going to propel the ions through the channel. And then once it's through the channel, it'll be rehydrated with the water molecules that are available inside the cell. So this is thousands of times faster than transport through transport proteins. So if we want to look at how the ion channels achieve their selectivity, what's going to happen is that there is a desolvation energy associated with removing the water molecules from the hydrated potassium ion sphere prior to binding in the ion channel. However, once the ion binds to the channel site, the amino acids are located in such a position that it's going to mimic the original hydrated potassium ion. So the resolvation energy that we achieve upon binding to the site inside the channel is sufficient enough to overcome the desolvation energy that was lost upon removing the water molecules. If we want to contrast that with sodium, if sodium were to try to get through the potassium ion channel, what happens here is we also have that desolvation process that occurs with the sodium ion prior to binding to the binding site in the channel. If these ions are hydrated, they're too large to fit through the channel, so that water's got to be removed at the start. However, since the sodium ion is much smaller than potassium, you don't get maximal kind of resolvation when that sodium ion binds to the potassium ion binding site. Therefore, this favorable resolvation energy that we regain isn't sufficient to overcome all of that desolvation energy we lost upon removal of the water molecules. And therefore, you're going to see much higher selectivity of the channel for the larger potassium ion compared to the sodium ion. Now, our ionophores are going to work very similar to the ion channels, except in this case, these are going to be really small, almost mini portable ion channels, where the ionophore is going to surround this is a potassium valinomycin. Valinomycin is the ionophore complex, so that's going to mimic the hydrated potassium ion, giving it that favorable energy that's going to allow it to transport potassium across the cell membrane. And again, remember that these are going to go in whichever direction is preferred by the electrochemical gradient. So one last slide here to try to summarize most of what we've talked about. Starting at the top with the transport of glucose across the cell membrane. So in this example, the glucose is going down its concentration gradient. So this is an example of facilitated diffusion. We do not need any additional energy source. Once the glucose gets inside the cell, it can undergo glycolysis through the citric acid cycle, oxidative phosphorylation, resulting in an increase in the concentration of ATP. This increased concentration of ATP causes the ATP-cated potassium ion channel to close. And when this occurs, we are going to then open the calcium ion channel, leading to an increase in our intracellular calcium ion concentrations. That then allows for the granules containing insulin to go to the surface of the membrane, where we can get insulin secretion through exocytosis. Now, just to point out, we did use, in this case, ATP in order to close the potassium ion channels. However, this is not an active process. Remember, if we have something that's gated, it's just the presence of the ATP causes the gate to close. The ATP is not hydrolyzed or consumed, so this would still be going. That process is not an active process. It only occurs based on the concentration gradients. The other thing I'd like to bring in here would be the valinomycin, which is the potassium ionophore. So recall that ionophores transport ions going down or with the concentration gradients. So in this example, the presence, if we added valinomycin into the mix here, its presence would have the same effect as opening the potassium ion channels. So if we were to add valinomycin, what we would then expect is to have a decrease in the secretion of insulin, because it will essentially have the same effect as opening that potassium ion channel. So that about wraps it up and summarizes everything you need to know about membrane transport.